The cell is frozen and then tapped to cause it to break. The lipids of the membranes are weak when frozen, and thus, the fracturing often follows the membranes, separating one lipid layer from the other. Here the fracture passed irregularly through the cytoplasm and then entered the nuclear envelope. The nuclear envelope consists of the outer and inner membranes, with a space separating them. The two membranes fuse together at the nuclear pores. Nuclear pores may be distributed uniformly over the nuclear surface or may occur in bands or patches in some species. Actually, we do know, now in 2016. The model plant Arabidopsis thaliana has 135 million bits of information. Each page in this book can hold 7,670 characters, so if the genome sequence of a thaliana were written on these pages, we would need 17,601 pages. Loblolly pine, Pinus taeta, has much larger nuclei with more information, and for that we would need 2,627,118 pages. Plants do things differently. Box 3-2 calcium, strong bones, strong teeth, but not strong plants. Most plants and animals need hard parts. Wood is strong enough to support the weight of a tree, and bones play a similar role in animals. Seeds are often protected by resistant shells such as those of walnuts and almonds, and animal shells protect clams and oysters. Our teeth are so tough that they can chew through almost anything. Although plants and animals use hard parts for similar roles, plants rely on thick, tough cell walls, whereas animals use calcium salts. Would it be possible for plants to use bone-like material? We can analyze this as a set of alternatives and their consequences. The present alternative, wood, consists of cellulose and a chemical called lignin. Both are carbohydrates that a plant itself makes through photosynthesis, and thus, they are readily available. And both are remarkably inert, having little impact on other aspects of the plant's metabolism. In contrast, calcium and its salts participate in many metabolic pathways, and building or resorbing shells, bones, or teeth has a broad impact on cell physiology. Shells consist of calcium carbonate, and as animals use carbonate ion to build a shell, the acidity of the protoplasm is altered. Furthermore, animals can digest part of their shells if they need the calcium elsewhere, and this liberation of carbonate will again affect the pH. This is tolerable for marine organisms because they use carbonate from the surrounding seawater rather than from their own protoplasm, so their pH is not affected. If the shell is resorbed later, the liberated carbonate is likewise dumped outside the animal into the seawater. Animals like us, with an internal skeleton, use calcium phosphate in our bones and teeth. Calcium carbonate's tendency to alter pH is too dangerous for us, and our skeleton cannot use seawater as a carbonate reservoir. The phosphate ion that we use has little effect on a cell's acidity, and, furthermore, if we resorb bone for some reason, the liberated phosphate is a resource for many other metabolic reactions. It is not a liability at all. Figure B3, 2 a leaf clearing of maidenhair tree, showing several red-stained leaf veins that conduct sugars out of the leaf. Such veins are the targets of aphids and other sucking insects. The same tissue, in polarized light. A cross-section of Aristolochia wood, crystals are present in the two bands of tissue with blue-stained walls, see. This is a soft tissue in wood and is the site where sugars and other nutrients are stored. The same tissue as, but with polarized light. Plants too must be careful with calcium and its salts. Calcium carbonate's disturbance of cell acidity is just as dangerous for plants as it is for us, but plants do not use calcium phosphate either, perhaps because it requires too much phosphate. Most soils have just marginal amounts of phosphate and not quite enough to let plants grow optimally. If plant cells had walls made of bone, they would need much more phosphate than is available in soil. Like animals, plants must carefully control the concentration of dissolved calcium within their cells. The concentration must be kept at extremely low levels, otherwise, it would Interfere with metabolism. Roots block the entry of calcium, but so much leaks in accidentally that the plant must get rid of the excess somehow. Plants crystallize the calcium by producing oxalic acid. Calcium and oxalic acid react to form calcium oxalate, which is insoluble and forms crystals. Crystallized calcium is more or less inert and cannot interfere with the plant's metabolism. Calcium oxalate forms only tiny individual crystals rather than big bone-like structures. Wood with lignin is not as strong as bones or teeth, but its synthesis does not affect cell acidity. Plants can make all of the wood they need without relying on rare minerals from the soil. Plants and animals may do things differently, but there are sound biological principles underlying the differences. Inside every nucleus is one, two, or rarely several bodies called nucleoli, areas where the components of ribosomes are synthesized and partially assembled. Each ribosome contains a large amount of ribosomal RNA copied from ribosomal genes in the chromatin. Nuclei are large, complex organelles, and they occupy a major fraction of the volume of small dividing cells. However, most cells enlarge greatly after they stop dividing and become mature. As cells expand, the nucleus becomes a smaller percentage of cell volume. In certain conducting cells, the nucleus breaks down during cell differentiation, and the mature cell functions for several weeks or months without a nucleus. Our red blood cells are also enucleate at maturity. Both of these cell types are exceptional. They are enucleate only while performing a very limited type of metabolism, and they die shortly after losing the nucleus. Several types of plant cells are multinucleate. Central vacuole. Within young, small cells are organelles, vacuoles, that have just a single membrane, the vacuole membrane, also called the tonoplast. Vacuoles often appear to be empty because they store mostly water and salts that cannot be preserved for microscopy. However, they sometimes contain visible crystals, starch, protein bodies, and various types of granules or fibrous materials in addition to water and salts. As a cell grows and enlarges, vacuoles expand and merge until there is just one large central vacuole. Because it contains primarily water and salts, the central vacuole can expand rapidly, forcing the cell to grow rapidly as well. Animal cells must synthesize complete protoplasm to grow, but plant cells need only increase the amount of vacuole or water. For example, as flower buds open, petals expand from tiny to full-sized in just a few hours by simply enlarging their vacuoles, not by making new cells. Over a long period, plants must produce additional proteins, membranes, and organelles or else they would become almost pure water. In addition to cell growth, the central vacuole functions in storage of both nutrient reserves and waste products. In seed cells, vacuoles may be filled with protein that will be used when the seed germinates, perhaps 10 to 50 years after the material was deposited in the vacuole. Calcium regulates the activity of many enzymes, and plant cells keep protoplasmic calcium concentrations at the proper level by moving calcium into the vacuole, where it reacts with oxalic acid and crystallizes into an inert form. Other nutrients such as potassium may move in and out of the vacuole on a daily basis. The water-soluble pigments in many flowers, fruits, and red beets occur in vacuoles as well. Figure 3, 14 nucleoli usually stain more intensely than the rest of the nucleus. Occasionally, they stain a distinct color. Root tip of hyacinth. Often abbreviated PI. A system to excrete wastes never evolved in plants. Instead, metabolic waste products are pumped across the vacuole membrane and stored permanently in the central vacuole. The tonoplast is otherwise impermeable to these wastes, therefore, they cannot leak back into the cytoplasm where they would be harmful. Holding waste inside forever does not sound like an optimal situation, but it actually is selectively advantageous. Because most of these compounds are noxious and bitter, they deter animals from eating the plants. Mutations that result in excretion might make the cells taste good, which would be selectively disadvantageous. Figure 3:15 Azalea flower buds open overnight, the petals and stamens expanding rapidly as each cell fills its central vacuole with water. The flower maintains its shape by hydrostatic pressure, central vacuoles pressing cytoplasm firmly against cell walls. The central vacuole is a digestive organelle as well. As other organelles age and become impaired, they fuse with the tonoplast and are transported into the central vacuole, where digestive enzymes break them down. The liberated monomers are transported back into the rest of the cell, where they are used again. In animal cells, which do not have central vacuoles, this task is carried out by small vacuoles called lysosomes. Cytoplasm. If the nucleus and vacuole are excluded from the protoplasm, the remaining material is referred to as cytoplasm and contains the following structures. Mitochondria. Cells store energy as highly energetic but fairly unreactive compounds, such as sugars and starches. To use the energy, such compounds must be broken down and their energy must be used to synthesize new compounds that are both highly energetic and very reactive, the most common being adenosine triphosphate. Because these reactions involve high-energy, reactive compounds, they occur more safely within a discrete organelle than freely in the cytoplasm, where the intermediates might accidentally react with other cell components. Mitochondria are the organelles that carry out this cell respiration. Figure 3:16 The central vacuoles of these petal cells contain dissolved pigments, making the vacuoles visible. The pale area between each oval of pigment is intercellular space, cell walls, nuclei, and all of the protoplasm other than vacuole. Many steps of respiration are mediated by enzymes bound to mitochondrial membranes. The enzymes are located adjacent to each other so that the product of one reaction is passed directly to the next enzyme. This controls the highly reactive intermediates. Mitochondrial membranes are folded, forming large sheets or tubes known as cristae. This folding provides room for large numbers of enzymes. Reactions that do not involve highly reactive intermediates take place in the liquid matrix between the cristae. Around this complex of cristae and matrix is a second membrane, the outer mitochondrial membrane, which gives shape and a little rigidity to the mitochondrion. The outer membrane is rather freely permeable, but the inner mitochondrial membrane, which forms the cristae, is selectively permeable and has numerous pumps and channels. Mitochondria have their own DNA and ribosomes, both of which are different from those of the rest of the cell. Mitochondrial DNA is a circular molecule and lacks histones. The ribosomes are small and resemble those found in prokaryotes. Mitochondria are dynamic organelles. They can grow larger either as the cell does or as the cell's need for respiration increases. They are often about 1 micrometer in diameter but up to 5 μm in length and are even much longer in some cases. Mitochondria can also divide into two, increasing the number of mitochondria per cell. Under rare conditions, the mitochondria in some species fuse together, forming a single giant mitochondrion in each cell. Mitochondria occupy between 1% and 25% of the cell volume. The higher values are similar to those for animal cells. Because of their ability to divide and fuse, the actual number of mitochondria per cell may not be as important as their volume. Plastids. Plastids are a group of dynamic organelles able to perform many functions. One prominent activity is photosynthesis, carried out by the green plastids, chloroplasts. Diverse types of metabolism occur in other classes of plastid: synthesis, storage, and export of specialized lipid molecules, storage of carbohydrates and iron, and formation of colors in some flowers and fruits. Plastids are the site of synthesis of amino acids, isoleucine, valine, and those that contain aromatic rings, or are derived from aspartate. Each metabolism is associated with a particular type of plastid. As an organ changes, its plastids may also change, extensively altering their membranes and proteins. Plastids are found in all plants and algae but never occur in animals, fungi, or prokaryotes. Figure 3.17. The inner mitochondrial membrane is folded into plate-like cristae, giving it a large surface area. Many respiratory enzymes are intrinsic proteins embedded in the crista membrane. The large surface area makes it possible to contain many copies of each enzyme, and more reactions can occur simultaneously. Figure 3.18. Many enzymes must work together. The product B of enzyme 1 is the reactant of enzyme 2. A. If the enzymes are in solution, they may not be close together, and B must diffuse at random until it encounters enzyme 2. In the meantime, it may accidentally react with something else. B. If they are located side by side on a membrane, enzyme 1 can pass B directly to enzyme 2, not only speeding up the overall reaction but also eliminating the chance of accidental reactions. Like mitochondria, plastids always have an inner membrane and an outer membrane and an inner fluid called stroma. Plastids also have ribosomes and circular DNA that is not associated with histones. Plastids grow and reproduce by pulling apart. Plastids of young, rapidly dividing cells are called proplastids and are very simple. The inner membrane has a few folds but little surface area. When exposed to light, proplastids develop into chloroplasts, which are green owing to the presence of the photosynthetic pigment chlorophyll. Because many intermediates of photosynthesis are highly reactive, energetic compounds, the controlling enzymes must be incorporated into the membranes, just as in mitochondria. This requires the inner membrane to become more extensive and elaborately folded. Membrane sheets, thylakoids, project into the stroma. This increase in membrane area also provides more space for the insertion of the photosynthetic pigments. Chlorophyll has a lipid-soluble tail and, thus, is part of the membrane. In certain regions, the thylakoids form small bag-like vesicles that stack together. The stack of vesicles is called a granum. A key feature of photosynthesis is the active transport of protons into a small space to build up an electrical charge. The grana vesicles are needed to accumulate these protons from the stroma. The actual conversion of carbon dioxide to carbohydrate occurs in the stroma, catalyzed by enzymes free in solution rather than bound to any of the membranes. Figure 3.19. All plastids have an outer and an inner membrane, but in chloroplasts, the inner membrane is extensive and highly folded. The single membranes are thylakoids, and the multiple membranes are actually stacks of flattened thylakoid vesicles. The stacks are called grana. Like mitochondrial cristae, the extra surface area of the inner membrane provides room for many copies of each enzyme. The photosynthetic pigment chlorophyll is part of the membrane lipid layer. The grana are interconnected by thylakoid membranes, and the liquid stroma surrounds the grana. Because the thylakoid membranes are selectively permeable, the concentrations of chemicals inside the thylakoid space differ from those in the stroma. This is essential for photosynthesis. Table 3, 7 types of plastids. Chloroplasts are larger than mitochondria, approximately 4 to 6 micrometers in diameter. An individual leaf cell may contain as many as 50 chloroplasts. When chloroplasts photosynthesize rapidly, they produce sugar faster than the cell can use it, and thus, it is temporarily polymerized into starch grains inside the chloroplasts. In plant tissues that cannot photosynthesize, proplastids develop into amyloplasts, which accumulate sugar and store it as starch for months or years, as in starchy seeds like wheat, rice, corn, or vegetables such as potatoes and yams. Each amyloplast produces large starch grains that virtually fill the stroma. Few internal membranes are present. In starchy vegetables, amyloplasts constitute the bulk of the tissue. If exposed to light, amyloplasts can be converted to chloroplasts. Non-photosynthetic parasitic plants such as Canophilus lack chloroplasts but do have amyloplasts. No plant is ever without some form of plastid. In some flowers and fruits, for example, tomatoes and yellow squash, bright red, yellow, or orange lipids accumulate in plastids as they differentiate into highly colored chromoplasts. An extensive, undulate system of membranes is present, but no grana, and the pigments may be present either as part of the membrane or as discrete droplets, plastoglobuli. As fruits ripen, chloroplasts often synthesize large amounts of lipid pigments, alter their thylakoids, and convert to chromoplasts, as when apples and tomatoes change from green to red. Lipid pigments are present in low amounts as small plastoglobuli even in leaf chloroplasts, but they are masked by the abundant green chlorophyll. They can be seen in autumn when cold weather causes chlorophyll, but not lipids, to be broken down and the leaves turn red or yellow. Many cells have large, unpigmented plastids that have neither chlorophyll nor lipid pigments. These leucoplasts may be involved in various types of synthesis. Many types of fats and lipids are synthesized only in plastids and then transported to other organelles and inserted into their membranes. Leucoplast is a purely descriptive term. Any colorless plastids, including perplastids and amyloplasts, can be considered leucoplasts. Iron is an essential nutrient for plants and animals and is stored attached to a large protein. Although almost identical to human ferritin, the plant protein is called phytoferritin. Ferritin is found throughout animal cytoplasm and nuclei, but phytoferritin is stored almost exclusively in plastids. As much as 80% of the iron in leaves may be found in chloroplasts, and the leucoplasts and amyloplasts of seeds often have especially large amounts. Ribosomes. Immersed in the protoplasm are ribosomes, particles responsible for protein synthesis. They are complex aggregates of 3 molecules of RNA and approximately 50 types of protein that associate and form 2 subunits. Compared with animal cells such as those in the liver and pancreas, most plant cells synthesize little protein and have few ribosomes. However, some do produce large amounts of proteins and are rich in ribosomes, such as the protein-rich seeds of legumes including peas and beans, and the cells that secrete the digestive enzymes of insectivorous plants. Each molecule of messenger RNA is long enough for 6 to 10 ribosomes to attach to it and read it simultaneously. The ribosomes are thus bound together by the messenger RNA, forming a cluster called a polysome. Figure 3, 20 cells from a developing bean seed. The large pink bodies are starch grains, the small bluish-red ones are protein bodies. These colors are not natural. The material was stained to make the colorless starch and protein visible. The starch grains occur in plastids called amyloplasts. As the bean germinates, the starch is used as a source of energy. When the starch decreases, the amyloplasts convert to chloroplasts and turn green. Starch grains that have been photographed in polarized light rather than being stained. Substances that have an orderly, crystalline structure shine brightly in polarized light. Freehand section of fresh potato. Each cell is filled with numerous oval starch grains. The thin walls of these cells are just barely visible. Figure 3, 21. Each tiny orange dot is a chromoplast. This is from an intensely bright orange pepper. Just a few chromoplasts provide brilliant color. Chromoplasts are located in the periphery of the cell because the central vacuole occupies most of the cell center. The largest cell appears to have chromoplasts everywhere, but we are just viewing the nearest wall, not the center of the cell. Many chromoplasts are aggregated around the nucleus. Endoplasmic reticulum. A typical plant cell is so small that diffusion, the random movement caused by molecular motion, carries a molecule to many different parts of the cell every second. Diffusion may be the only means by which small molecules like monosaccharides and cofactors move around the cell, but some large molecules such as proteins are carried by the endoplasmic reticulum, a system of narrow tubes and sheets of membrane that form a network throughout the cytoplasm. A large proportion of a cell's ribosomes are attached to the ER, giving it a rough appearance. Consequently, this ER is called rough ER. As an attached ribosome synthesizes a protein, it passes through the membrane and collects in the lumen. If the protein is a storage product, as in seeds of legumes, it merely remains in the ER, which may become quite swollen. However, if the protein is to be secreted, digestive enzymes adhesive proteins, then its accumulation causes regions of the ER to form vesicles. These detach, move to the plasma membrane, and fuse with it, releasing their contents to the cell's exterior by exocytosis. In many cases, the protein must be modified before export. If so, the ER pinches off only very small vesicles from regions where the ER is close to another organelle, the dictyosome, which carries out the protein modification. Figure 3.22 ribosomes only rarely occur free in the cytoplasm. Instead, they are usually attached to membranes such as the type shown here, called endoplasmic reticulum. The amount of ribosomes attached is greater in cells that produce abundant protein and less in cells that synthesize little protein. Electron micrograph of a cross-section of a hair cell that secretes protein-rich mucilage. The mucilage is synthesized in the ER, which is unusually abundant for a plant cell. ER that lacks ribosomes is smooth ER, and it is involved in lipid synthesis and membrane assembly. The lipids range from simple to extremely complex. As they are produced, they are inserted into the membrane, and then vesicles form and pinch off, carrying the new membrane to other parts of the cell. After the ER-derived vesicles reach the correct organelle, they fuse with it, and the vesicles become a new patch of membrane in the organelle. SER is abundant only in cells that produce large amounts of fatty acids, oils, and fragrances of many flowers. Dictyosomes. Much of the material secreted by a cell must first be modified by a dictyosome, a stack of thin vesicles held together in a flat or curved array. ER vesicles accumulate on one side of the dictyosome and then fuse together and form a wide, thin vesicle called a cisterna that becomes attached to the dictyosome. Soon more ER vesicles gather next to this one and form a new cisterna. The first cisterna becomes embedded more deeply in the dictyosome as more vesicles accumulate on that side, which for obvious reasons is the forming face. At the other side, the maturing face, vesicles are being released, their contents have been processed. After separation, vesicles can move to the plasma membrane and release their contents. The outer edges of dictyosomes form an interconnected network of curving tubes, and these may absorb the contents from the center of the dictyosome cisterna and then detach and move away. It is not known why some dictyosomes concentrate material in the central cisternae, whereas others use the peripheral ones. Dictyosomes can form large, complex associations. In animal cells that secrete very large amounts of protein, hundreds of dictyosomes associate side by side and form a cup-shaped structure called a Golgi body or Golgi apparatus. In the Golgi body, the dictyosomes' maturing faces are on the inner side of the cup, whereas the forming faces and associated ER are on the outside. Dictyosomes only rarely aggregate into Golgi bodies in plants, one example being in root hairs. All of the dictyosomes are part of a giant Golgi body located at the tip of the hair where growth and cell wall formation occur. Different types of processing may occur within a dictyosome: modification of the vesicle's membrane or modification of its contents. If the vesicle is to fuse with the plasma membrane after release, the vesicle membrane must be made similar to the plasma membrane. If the vesicle is to remain separate from all other organelles, acting as a storage vesicle, its membrane must be made unique and incapable of fusion. Alteration of the vesicle's contents involves addition of sugars to proteins, forming glycoproteins. Sugar-containing proteins occur in the plasma membrane, the cell wall, and as storage products in seeds. Strong evidence is accumulating that dictyosomes also polymerize sugars into polysaccharides used in cell wall construction. Movement of vesicles from ER to dictyosomes and then to other sites was hypothesized on the basis of the presence of numerous vesicles located between ER and dictyosomes and the similarity of the contents of dictyosome cisternae to those of vesicles found fusing with the plasma membrane. This hypothesis predicts that if a secretory cell is given a very brief dose of radioactive sugar or protein precursors, much of the radioactivity is soon found in the ER and later in the dictyosomes. Even later, both are non-radioactive, but vesicles at the plasma membrane and external material are radioactive. Experiments have verified both this prediction and the related hypothesis that new membrane is synthesized in the ER and then transported by vesicles to growing organelles. Although organelles appear to be distinct entities when viewed by light or electron microscopy, they are actually highly interrelated by this membrane flow. All membranes of the cell, except the inner membranes of mitochondria and plastids, actually constitute just one extensive system, the endomembrane system. Figure 3.23 with rapid dictyosome activity, as in these mucilage-secreting cells, the cytoplasm may almost fill with dictyosome vesicles, and fusion of vesicles with the plasma membrane is so abundant that the plasma membrane appears scalloped. Face view of dictyosome isolated from a plant cell, the network of peripheral tubules is visible. Figure 3.24 vesicles derived from ER migrate a short distance to a dictyosome-forming face. At the forming face, the vesicles fuse into a new dictyosome vesicle, called either a vesicle or a cisterna. The cisterna moves through the dictyosome as more vesicles form on one side while other vesicles are released from the maturing face. Figure 3.25 This is a mucilage cell. Mucilage-filled dictyosome vesicles secreted mucilage outside the protoplast. Mucilage could not penetrate the cell wall, so as it accumulated, the protoplast had to decrease in volume by removing water from the central vacuole. The cell died, and now the cell consists only of cell wall and mucilage. In a few cases, it has been possible to measure the rate of membrane flow. The insectivorous plant Drosophilum has leaves covered with glands that produce a sticky secretion. The fluid contains digestive enzymes that are processed by dictyosomes in the gland cells. After an insect has been caught, each gland produces a visible drop of digestive fluid at a rate of 1.3 micrometers per minute. Dictyosome vesicles are about 0.15 micrometers in radius, thus, each has a volume of 0.014 micrometers, and approximately 100 must fuse with the plasma membrane every minute to deliver the 1.3 micrometers of fluid and enzymes. Each vesicle has a surface area of 0.27 micrometers, so 27 micrometers of vesicle membrane fuses with the plasma membrane every minute during secretion. From calculations of the average cell volume and surface area of the plasma membrane, these secretory cells could double their plasma membrane every 20 seconds. It is important for these cells to be able to retract membrane material from the plasma membrane and recycle it. The mucilage-secreting cells of root tips are more leisurely. Each cell has approximately 800 dictyosomes that together contribute 14 to 26 micrometers of membrane per minute to the plasma membrane, which has a surface area of about 1,000 micrometers. Each dictyosome receives a new cisterna every 20 to 40 minutes. Microbodies. Viewed by electron microscopy, cells are seen to contain numerous small, spherical bodies approximately 0.5 to 1.5 micrometers in diameter. These are so nondescript that it is not easy to tell exactly what they are: ER vesicles, dictyosome vesicles, or distinct organelles. However, by using special chemical reactions and stains, it has been possible to determine the contents and even the types of reactions in these bodies. Some are organelles now called microbodies, and there are two classes, peroxisomes and glyoxysomes. Both types isolate reactions that either produce or use the dangerous compound peroxide, HO. If peroxide were to escape through the microbody membrane, it would damage almost anything it encountered. However, both types of microbody contain the enzyme catalase, which detoxifies peroxide by converting it to water and oxygen. Botany and beyond. Box 3-3 The metric system and geometric aspects of cells. The metric system. In 1791, the French Academy proposed that a new system of measurement be adopted to simplify and regularize weights and measures. This metric system is used not only for all scientific measurements but also for engineering, commercial, and ordinary purposes in every country of the world except the United States. See the conversion tables at the end of this box. The fundamental unit is the meter. However, for long distances, the kilometer is used, and for small objects, centimeters, millimeters, and so on are more convenient. Volumes are also based on the length measurements, and weights are based on the gram, the weight of 1 centimeter of water. The metric system has two advantages: measurements in objects made in one country can be understood or used immediately in any other country, except the United States, and measurements in one unit can be converted easily to other units. 35,645 meters is 35.645 kilometers, but 35,645 feet is how many yards or miles? 1 kilometer equals 1,000 meters, 1 meter equals 1,000 millimeters, 1 millimeter equals 1,000 micrometers, 1 micrometer equals 1,000 nanometers, 10 nanometers equals 10 micrometers equals 10 millimeters equals 10 m equals 10 kilometers. These are based on multiples of 1,000. The centimeter is only 1/100 meter or 10 millimeters, but it is such a convenient size for measuring plants and animals that it is used even though it disrupts the otherwise perfect regularity of increments of 1,000. Geometry of cells. A quantitative understanding of plants provides extremely valuable insight into their biology. The following formulas are simple but will allow you to think about plants more precisely. Area of a rectangle equals length width. Area of a triangle equals 1/2 length width. Volume of a cubic space equals length width height. Diameter of a circle equals 2 radius. Circumference of a circle equals 2 pi radius. Area of a circle equals pi radius. Surface area of a sphere equals 4 pi radius. Volume of a sphere equals 4 pi 3 radius. Volume of a cylinder equals area of base height equals pi radius 2 height. Surface area O F a cylinder equals circumference of circle height plus ends equals pi diameter height plus ends. Many plant cells are almost cube-shaped and have edges about 20 micrometers long. The volume of such a cell is the volume of a cube. Equals LWH equals 20 micrometers 20 micrometers 20 micrometers equals 8,000 micrometers. Do not forget to multiply the units to get μm. Now consider the nucleus of the cell. Many plant nuclei have a diameter of about 8 micrometers, and thus, their radius is 4 micrometers. The volume of a nucleus is V equals 3 is equal to 4.18864 micrometers equals 268 micrometers. The nucleus, although an important part of a cell, may not be a large part. In our average cell of 8,000 micrometers volume, the proportion that is nucleus is 100% equals 268 micrometers. 8,000 micrometers 100% equals 3.3%, a very small part of the cell volume. Mitochondria are about 1 micrometer in diameter and are quite variable in length. Let us assume that we have measured many and found the mean length to be 5 micrometers. Mitochondria are almost cylinders, so we can use that formula to calculate their volume. Figure B3, 3a dimensions of a hypothetical cell. Notice that the mitochondrion is not drawn to scale. Figure B3, 3b. Most of us have a set of standard metric weights readily available. A dollar bill weighs exactly 1 gram, 2 pennies weigh 5 grams, 15 quarters and 3 pennies weigh almost exactly 100 grams, and a small post-it note weighs 1/10 of a gram. Figure B3, 3c. Many rulers have centimeters along one side and inches along the other. Volume of a cylinder equals area of a circular cross-section height equals 0.7852 micrometers equals 3.93 micrometers. Mitochondria often constitute about 7.5% of a cell's volume. For our typical cell, that would be 8,000 micrometers. 0.075 is equal to 600 μm. How many actual mitochondria would there be in each cell? If each mitochondrion has a volume of 3.93 micrometers, the number is total. Volume equals 600 micrometers. 3.93 micrometers equals 153 mitochondria in each cell. The surface of a cell or organelle is the space through which material enters and leaves the object by diffusion, facilitated diffusion, or active transport. Objects with large surface areas can absorb or lose material more rapidly than those with smaller surface areas. Also, surface area is a measure of the room available for placing membrane-bound enzymes and other intrinsic membrane proteins. In our hypothetical cubical cell, each surface has an area of 20 micrometers. 20 micrometers equals 400 micrometers. A cube has 6 sides, and thus, the total surface area of the plasma membrane is 400 micrometers equals 2,400 micrometers. That is about 1/30 of the surface area of the period at the end of this sentence. The nucleus of the cell has a surface area of 4 pi equals 12.5616 micrometers equals 201 micrometers, less than 1/10 that of the cell. The cylindrical mitochondria each have a surface area of their outer membrane of 3.1411 micrometer. 5 micrometers equals 15.7 micrometers. What, however, of their inner membrane? Assume that measurements reveal that each crista is a cylindrical tube 0.2 micrometers wide and 0.9 micrometers long, and each mitochondrion contains an average of 45 cristae, then the surface area of each is 3.1410. 0.2 micrometers. 0.9 micrometers equals 0.565 micrometers. The 45 in each mitochondrion have a total surface area of 45. 0.565 micrometers equals 25.4 micrometers available for respiratory enzymes. The 177 mitochondria of the cell would have a total inner surface area of 4,496 micrometers, greater than that of the plasma membrane area. Thus, folding the inner membrane into cristae provides a large amount of extra surface area for enzymes. Figure B3, 3D. The metric system measures volumes in liters and milliliters. A tablespoon is 14.7 milliliters, and a teaspoon is exactly 5 milliliters. 1 cup is slightly less than 250 milliliters. Drops of milk are irregular, but 20 drops of water equal 1 milliliter. The measuring cup and the beaker use the abbreviation ml, but the correct abbreviation is ml. Figure 3, 26 electron micrograph showing 2 chloroplasts with 2 peroxisome-type microbodies next to them. In many plants, photosynthesis is accompanied by a process called photorespiration, in which peroxisomes produce the amino acid glycine. Peroxisomes are involved in detoxifying certain byproducts of photosynthesis and are found closely associated with chloroplasts. In animals, peroxisomes are abundant in liver and kidney cells, where they break down foreign compounds that contaminate our food. Glyoxysomes, which occur only in plants, are involved in converting stored fats into sugars. They are important during the germination of fat-rich, oily seeds such as peanut, sunflower, and coconut. All the previously mentioned, except ribosomes, are organelles composed of membranes. The following are non-membranous organelles. Cytosol. Most of the volume of cytoplasm is a clear substance called cytosol or hyaloplasm. It is mostly water, enzymes, and numerous chemical precursors, intermediates, and products of enzymatic reactions. Within cytosol are free ribosomes, as well as skeletal structures, the microtubules and microfilaments. Microtubules. Microtubules are the most abundant and easily studied of the structural elements of a cell, and they have many functions. They act as a cytoskeleton, holding certain regions of the cell surface back while other parts expand. Without them, cells would be just spheres, but by reinforcing specific areas, cell growth and expansion are directed to weaker areas. In other cases, microtubules assemble into arrays like an antenna which either catch vesicles and guide them to specific sites or cover a region, thereby excluding the vesicles. Finally, microtubules are the means of motility for both organelles and whole cells. The framework that moves chromosomes during division of the nucleus is composed of microtubules, and microtubules can attach to and move whole nuclei, mitochondria, and other organelles. Table 3, 8 subunits of the cell. Figure 3, 27 microtubules that are part of a dividing nucleus and are involved in pulling chromosomes to the ends of the cells. Alpha and beta tubulin associate into a dimer called tubulin, and dimers aggregate into a microtubule. When no longer needed, microtubules depolymerize back to the monomers, which are recycled to build other microtubules. Figure 3, 28 microtubules are often located next to the plasma membrane. They may act as a screen that keeps vesicles away from the plasma membrane, or they may pull the plasma membrane away from the cell wall, allowing material to accumulate there. Microtubules are composed of two types of protein with a globular tertiary structure, alpha tubulin and beta tubulin. These associate as dimers called tubulin that further crystallize into a straight tubule with a diameter of 20 to 25 nanometers. This is a reversible process. When a microtubule is no longer needed, it depolymerizes back into its component monomers, which disperse into the cytosol until the cell needs to assemble a new microtubule. When microtubules occur as individuals or small clusters, new tubulin dimers are added to or removed from one end automatically. In other instances, microtubules occur in large clusters, often in a highly ordered arrangement, and a small body is usually associated with the orderly production of microtubules. For example, when the nucleus undergoes division, an array of microtubules called the spindle is formed in the middle of the cell, and spindle microtubules push and pull the chromosomes to their proper positions. In all animals and in some fungi and algae, a pair of organelles called centrioles is associated with the formation of the spindle. A centriole is made up of 9 sets of 3 short microtubules. The 9 triplets are held together by fine protein spokes. Centrioles were assumed to be responsible for the organization and polymerization of the spindle microtubules, even though plants never have centrioles. Much more elaborate sets of precisely arranged microtubules occur in cilia and flagella, which appear to be identical except that cilia are short and occur in groups, whereas flagella tend to be much longer and usually occur either singly or in sets of 2 or 4. Both are present on many types of algae and motile fungi, but in plants, only the sperm cells have flagella. In flowering plants and conifers, no cells, not even sperm cells, ever have cilia or flagella. Figure 3, 29. The two members of a pair of centrioles are always located perpendicular to each other. Here, one is seen in transverse section, the other in longitudinal section. Each part of a centriole contains a circle of 9 sets of 3 microtubules attached to each other by fine filaments along their sides. Figure 3, 30 flagella occur on many algal and fungal cells, especially the unicellular organisms. This is the alga dunaliella. Most cells of multicellular organisms are not flagellated, but their sperm cells are. Figure 3, 31 cilia and flagella are composed of 2 central single microtubules surrounded by 9 sets of 2 microtubules. The doublets are not merely attached to each other, they actually share tubulin monomers. The outer member of each doublet has 2 short arms. In cross-section, each cilium or flagellum has a 9+2 arrangement. 9 pairs of fused microtubules surround 2 individual microtubules. The outer doublets each have 2 arms composed of the protein dynein, and each doublet is connected to the central pair of microtubules by protein spokes. The dynein arms convert the chemical energy of ATP into kinetic energy and bend, walking along the adjacent microtubule doublet. One set of doublets slides relative to the adjacent set, causing that side of the cilium or flagellum to become shorter and bend the structure. Then, as another set of microtubules slides, the cilium or flagellum bends in a different direction. The result is a powerful beating motion. If the cilia or flagella are on small organisms such as algae, fungi, or protozoans, the organism swims rapidly and gracefully. Cilia and flagella can polymerize and depolymerize rapidly, but they never form autonomously. Each is always associated with a basal body. Basal bodies appear to be identical to centrioles by electron microscopy. It was assumed that basal bodies organize the formation of flagellar microtubules, but recent studies have shown that as a flagellum grows, new monomers of tubulin and dynein are added to the tip, not the base where the basal body is located. The exact relationship between flagella and basal bodies is still not known. Microfilaments. Like microtubules, microfilaments are constructed by the assembly of globular proteins, in this case, just one type, actin. Microfilaments are narrower than microtubules, and they have been implicated in different types of structure and movement. Storage products. Many cells exist in an environment in which resources alternate between abundance and scarcity. To survive times of scarcity, cells must accumulate and store extra nutrients. Most often, the reserves consist of sugars that have been polymerized into starch in amyloplasts or converted into lipids and stored as large oil droplets in oily material like peanuts and sunflower seeds. However, with many other storage products, the function and advantages are not so obvious. Many plants store crystals of calcium oxalate or calcium carbonate, others accumulate large amounts of silica, tannins, or phenols. Because plants have no excretory mechanism, numerous waste products must be stored within the cells. Cell wall. Almost all plant cells have a cell wall, only sperm cells of some seed plants lack one. It is often regarded as simply an inert secretion, providing only strength and protection to the protoplasm inside. However, considerable metabolism occurs in the wall, and it should be considered a dynamic, active organelle. The cell wall contains a considerable amount of the polysaccharide cellulose. Adjacent, parallel cellulose molecules crystallize into an extremely strong microfibril 10 to 25 nanometers wide. Numerous microfibrils are wound around the cell, completely covering the plasma membrane. Each cellulose polymer grows only at one end, where a complex of enzymes adds new glucose residues, one molecule at a time. The enzymes are intrinsic proteins of the plasma membrane, and as they add new sugars to the chain, the enzymes float forward in the membrane. New cellulose molecules can be added only on the inner side of the wall, adjacent to the plasma membrane. Figure 3.32 Avocado contains very large numbers of oil bodies. This view was prepared merely by smearing a small amount of ripe avocado fruit on a microscope slide. Virtually every visible thing is an oil body. Other organelles are much less abundant. Figure 3.33 Calcium oxalate crystals. When calcium is part of a crystal, it is physiologically inactive and cannot affect membranes or the tertiary structure of proteins. These are cubic crystals in wood. Figure 3.34 This type of preparation is called a leaf clearing. A leaf was treated to make its tissues transparent, and then it was stained to reveal certain contents. The dark red, irregularly shaped vacuolar contents are tannins, which can denature proteins. They have a bitter taste and damage the mouth and stomach proteins of insects that try to eat them. The band running vertically through the micrograph is a leaf vein. The bark of a cactus. As its cells aged, they accumulated irregular masses of tannin that have stained red. Cellulose microfibrils are bound together by other polysaccharides called hemicelluloses, which are produced in dictyosomes and brought to the wall by dictyosome vesicles. Hemicelluloses are deposited between the cellulose microfibrils and bind to cellulose with hydrogen bonds, producing a solid structure that resembles reinforced concrete. In multicellular plants, the wall of one cell is glued to the walls of adjacent cells by an adhesive layer, the middle lamella, composed of a third class of polysaccharides, pectic substances. All plant cells have a thin wall called the primary cell wall. In certain cells that must be unusually strong, the protoplast deposits a secondary cell wall between the primary wall and the plasma membrane. The secondary wall is usually much thicker than the primary wall and is almost always impregnated with the compound lignin, which makes the wall even stronger than hemicelluloses alone can make it. Lignin resists chemical, fungal, and bacterial attack. Both primary and secondary cell walls are permanent. Once deposited, they are almost never degraded or depolymerized, as can be done with microtubules and microfilaments. Fungal cells. Cells of fungi are similar to plant cells, with two important differences: they do not contain plastids of any type, and their walls contain chitin, not cellulose. Chitin is physically similar to cellulose, being tough, inflexible, and insoluble in water, but it contains nitrogen and is synthesized by a different mechanism than that used for cellulose. Whereas plant cells tend to be box-shaped or fiber-like, fungal cells are often extremely narrow, long tubes with many, tiny nuclei. Figure 3:35. Several layers of cellulose microfibrils are visible, other layers are present deeper in the wall. Each layer provides strength in the direction parallel to the microfibrils. Other wall components have been dissolved away to reveal the cellulose. Cellulose synthesizing enzymes are large proteins embedded in the plasma membrane. Several proteins form a cluster called a rosette. Sugar monomers are absorbed by the enzymes of the membrane interface and then passed across the membrane and polymerized into cellulose on the outer face. The growing microfibril extends out from the rosette and lies against microfibrils already in place. Because the microfibril cannot push into the wall, the rosette is pushed through the membrane in the direction of the arrows as the microfibril grows. Figure 3:36. Although cellulose microfibrils lie close together, they do not bond with each other, and thus, a wall of pure cellulose is weak. Hemicelluloses and pectins are short, branched molecules that interact with several adjacent microfibrils, linking them together and forming a solid, three-dimensional mesh. The relative amounts of these components can vary, resulting in some walls that are flexible and others that are rigid, some strong and others weak. Associations of cells. In unicellular organisms, such as simple algae, protozoans, and most prokaryotes, each cell is a complete organism and does not interact directly with other cells. Such cells can communicate indirectly, at least to a small extent, by releasing chemicals that affect the other cells, but such interactions are rare, usually occurring only during the attraction of other cells during sexual reproduction. In multicellular organisms, each cell automatically, unavoidably interacts with its neighboring cells. They must share the same sources of photosynthate, oxygen, carbon dioxide, salts, and water. Whereas a unicellular organism can merely excrete its wastes across its plasma membrane, cells of a multicellular organism are not so free to do so. Just as important are cellular interactions by which cells not only sense that they are part of a larger organism but also identify which part they are and how they should differentiate. In a developing embryo, the proper cells must be instructed to begin forming an embryonic shoot while other cells are induced to form the embryonic root, vascular tissues, seed leaves, and so on. This requires extensive, sophisticated intercellular communication. One communication method is like that used by unicellular organisms. A cell secretes specific compounds that inform the surrounding cells of what it is doing metabolically and developmentally. A second method is connections between the cells. Direct physical contact between cells, as exists in animals, cannot occur in plants because the two primary walls and middle lamella are located between any two adjacent protoplasts. However, plant cells are interconnected by fine holes in the walls. A plasmodesma is only about 40 nanometers in diameter, but the plasma membrane of one cell passes through it and is continuous with the plasma membrane of the adjacent cell. A small channel of cytosol also passes through, as does a short section of specialized ER. Figure 3:37. Plasmodesmata are complexes that consist of fine holes in the primary walls. They also contain plasma membrane, liquid, and a tubule attached to the ER. Face view of a cell wall, showing an oval-shaped thin area, a primary pit field. The small open areas outlined by single cellulose microfibrils are holes where plasmodesmata were located. Protoplasm was removed during specimen preparation. The abundance of plasmodesmata in a wall is quite variable. They can occur singly or in clusters of 10 to 20 or more. In regions of clustered plasmodesmata, the two primary walls are often particularly thin, and the area is called a primary pit field. In tissues in which considerable transport occurs, many primary pit fields and plasmodesmata are present, but in regions where little movement of material happens, few plasmodesmata are found. Table 3:9 Frequency of plasmodesmata. Because plasmodesmata are actually cytoplasmic channels from one protoplast to another, the individuality of the cells is diminished. All of the protoplasm within a single plant is part of one interconnected mass, the symplast. In some of the nutrient-conducting tissues and also in some of the reproductive tissue, plasmodesmata are so large and numerous that the cells act more like one single large cell than like individual cells. In most tissues, however, any two adjacent cells act rather independently. The communication through the plasmodesmata is not enough to prevent individuality. The fact that walls keep protoplasts physically separated from each other has another consequence. Walls act as a second, non-living compartment inside plants. Water moves through cell walls by capillary action, just as it moves through tissue paper. In addition, many cells do not abut each other tightly but instead have intercellular spaces between them, at least at their corners. In some tissues, especially leaves, cells are so loosely connected that most tissue volume is intercellular space, less than half is actually symplast. These spaces plus the cell wall constitute the apoplast. The apoplast and the symplast together make up the entire plant. The apoplast acts as a series of channels and spaces that permit the rapid diffusion of gases, which is necessary because plants do not have lungs. Diffusion through a gas-filled space is approximately 10,000 times faster than through a liquid-filled space. Without an apoplast, large, bulky plant tissues such as tree trunks, tubers, and fruits would be impossible. The interior tissues would suffocate. Figure 3:38 In regions where cells press against each other, their surfaces form flat contact faces. Primary pit fields and plasmodesmata occur in contact faces. Notice that chloroplasts avoid contact faces and instead are located next to intercellular spaces, which are regions where they can obtain carbon dioxide. High magnification view of a contact face in face view. The white areas appear to be holes, but they are primary pit fields, areas where the walls are especially thin and plasmodesmata are numerous. If this same region were viewed with an electron microscope, each small white dot would look like figure 3:37b. The non-contact face is the region of cell wall that faces intercellular space rather than being pressed against an adjacent cell. At the next level. 1. Evolution of plastids and mitochondria. It is now relatively quick, easy, and inexpensive to sequence the DNA circles of organelles. We know the sequence and composition of every gene in the plastids of many plants, and more plastid genomes are being sequenced every week. Botanists are using this information to analyze the evolution of plastids. Algae in early plants had many more plastid genes, but in some plant groups, certain plastid genes have been transferred to the nucleus, unnecessary genes have been eliminated, and various rearrangements of genes on the plastid DNA circles are being studied. Through this we are learning how plastids and nuclear genes work together. Plant mitochondria are less well known, but they are being studied in the same way. Remember that nuclear genes are eukaryotic, whereas plastid genes are prokaryotic, being descended from cyanobacterial genes. 2. Organization and function of plant cell cytoskeleton. Plant cytoplasm is extremely dynamic. Cytoplasm moves from place to place in the cell, as do mitochondria, plastids, and nuclei. The cytoskeleton, composed of structural and motor proteins, is involved in this, but many details are only now becoming clear. The plant cytoskeleton appears to also be involved in the ability of a plant to detect that it has touched something, for example that a tendril has touched something and should begin to wind around it, or that a fly has landed on the trap of Venus flytrap. 3. History of our understanding of cells. Cells were discovered immediately after microscopes were invented in the 1660s. Two excellent books that describe the history of our knowledge about cell structure are The Birth of the Cell by Henry Harris and Views of the Cell, A Pictorial History by Joseph G. Gall. Summary. 1. All organisms are composed of cells, the fundamental units of life. Most cells contain the same types of organelles, and there are only a few basic types of cells. 2. In multicellular organisms, cells become specialized for specific tasks, making the entire organism more efficient. However, each cell then becomes dependent on the other cells of the organism. 3. Selectively permeable membranes and active transport are critically important for life. They make compartmentalization possible, in which an enclosed compartment has a chemical composition and metabolism distinct from those outside the compartment. 4. The outermost membrane of a cell is the plasma membrane. The protoplasm it surrounds is a protoplast. 5. Plant cells contain the following membrane-bounded organelles: nucleus, heredity and control of metabolism, vacuole, temporary or long-term storage, growth, mitochondria, respiration, plastids, photosynthesis, intracellular transport, lipid synthesis, dictyosomes, material processing, and microbodies, detoxification or lipid metabolism. 6. Plant cells contain these non-membranous organelles: ribosomes, protein synthesis, cytosol, the liquid matrix surrounding all other organelles, microtubules and microfilaments, skeletal structure, movement of organelles, cell wall, structure, cell shape, and various storage products such as crystals and lipid droplets. 7. Animal cells do not have a cell wall, plastids, glyoxysomes, or a central vacuole. Fungal cells do have a central vacuole, but they have no plastids and their wall is made of chitin, not cellulose. 8. A plant is composed of both its protoplasm and its intercellular spaces and walls. The apoplast can be important for diffusion of air and liquids. Important terms: apoplast, bilayer cell wall, centriole chloroplasts, chromoplasts, cilia, compartmentalization, cytoplasm, cytosol, dictyosome, endocytosis, endoplasmic reticulum, eukaryotic exocytosis, extrinsic proteins, flagella, fluid, mosaic membrane, freely permeable membrane, glycoproteins, Golgi apparatus, Golgi body, impermeable membrane, intercellular spaces, intrinsic proteins, lumen, microbodies, microfilaments, microtubules, middle lamella, mitochondria, molecular pumps, nucleolus, nucleus, organelles, plasmidesmata, plastids, primary pit field, prokaryotic ribosomes, selectively permeable membrane, symplast, tonoplast, tubulin vacuoles. Review questions. 1. In plants, each cell consists of a box-like surrounding a mass of, which in turn contains its own smaller parts, the. 2. In Table 3.1, the size of water-conducting vessel cells in oak are. Each millimeter contains 1,000 micrometers. Approximately how many of these cells could be laid end to end in 1 millimeter? Do you think such a cell would be visible if it were placed in your hand? How long are the fiber cells in hemp? How long is this in millimeters? How long is it in centimeters? Could you see one of these cells if it were in your hand? 3. Figure 3.1 is a light micrograph of a leaf cell, and it is typical of what you might see if you were to examine a moss leaf. What two cell structures can you see? 4. The division of labor among various types of cells in plants is important. Examine Table 3.2 and name the cells responsible for the following tasks: A. Collecting solar energy. B. Transporting water. C. Cell division and the production of new protoplasm. D. Water retention. E. Having pigments that attract pollinators. F. Producing poisons that inhibit animals from harming the plants. 5. Stone cells in coconut shells are part of the division of labor in plants. What modifications do they have that make it impossible for them to be dividing and growing cells? 6. All biological membranes are composed of and two layers of molecules. One of these components occurs as a thin solution only 2 molecules thick. A layer only 2 molecules thick is A. 7. Some of the proteins that are part of a cell's membranes are actually immersed in the membrane. Others are located outside the membrane and merely lie next to it. What are the names of each type? 8. Cell membranes are described as being fluid mosaics. What does that mean? Are all membrane components free to diffuse laterally anywhere within the membrane? 9. What are the 2 basic components of membranes? What are 3 ways that material can move from one side of a membrane to the other? Which method requires the plant to use energy? 10. Pieces of membranes frequently move from one area of a cell to another, but never as just flat patches, only as vesicles. Study Figure 3.9 and then draw examples of endocytosis and exocytosis using different colors for the 2 sides of the membranes. Notice that the black side of the membrane bilayer always touches the blue part of the cell in Figure 3.9. Can you come up with any type of vesicle fusion that would result in the black side of the membrane touching the green part of the cell? 11. Define each of these terms: a freely permeable membrane, an impermeable membrane, and a selectively permeable membrane. During your next exam, you may begin to sweat a solution of sodium, chloride, and water, but not proteins, sugars, or nucleotides. Are the membranes of your sweat gland cells freely permeable, impermeable, or selectively permeable? 12. Circle the correct answer. Plants, animals, fungi, and protists have cells. What is a key feature of eukaryotic cells? 13. The less frequently called the is the membrane that completely covers the surface of the protoplasm. It is, and it contains molecular pumps. 14. The nucleus serves as an archive for the organisms, all of which is stored as inside every nucleus. 15. Which organelle functions as storage of both nutrient reserves and waste products? What is the name of its membrane? 16. Which organelle carries out cell respiration? Each of these organelles has 2 membranes, a liquid portion and folded, sheet-like membrane structures. What are the names of these 4 components? 17. Plastids can grow and develop into many different types of structure. What is the name for the plastids that do each of the following? A. Carry out photosynthesis. B. Synthesize lipids and other materials. C. Store starch. D. Contain abundant colored lipids. E. Occur when tissues are grown without light. 18. Which organelle carries out protein synthesis? 19. What is the difference between RER and SER? SER is not very common in plants, but several examples are given in the text. What are they? 20. What is the role of dictyosomes in cell metabolism? What is their forming phase? Their maturing phase? 21. Draw a typical plant cell and include all of the organelles mentioned in this chapter. Most cells have specialized functions and increased proportions of certain organelles. How would you change your drawing if the cell is involved in photosynthesis? Is part of a yellow or orange petal? Secretes protein-rich digestive fluid? Needs a great deal of ATP? Stores starch? Has just grown a great amount? 22. Which of the organelles in question 21 are composed primarily of membranes? What are some of the ways that the membranes participate in the metabolic activities of those organelles? 23. Describe how material is brought to a dictyosome. What happens to material while it is in a dictyosome, and how is it released? What are some of the things that might happen to it after it is released? 24. What are alpha-tubulin and beta-tubulin? When they aggregate, what do they form? What structure in dividing cells is made with this substance? Algal, fungal, and animal cells move with structures made of this. What is the name of the structure? 25. Many animals and algae have cells with flagella, but do any plants have flagella? For example, do fern cells or moss cells have flagella? 26. How do microtubules act as a cytoskeleton? Are there times when they are particularly abundant? What are centrioles, basal bodies, and flagella? 27. Examine figures 3:32 to 3 to 34. What types of substances occur as storage products in plant cells? 28. What chemical do clams use to make strong shells? What do we use for our bones? Do plants use either of these chemicals in the construction of strong cell walls? What problem would plants have if they used either the shell material or the bone material? 29. In cell walls, adjacent, parallel cellulose molecules crystallize into an extremely strong 10 to 25 nanometers wide. These are bound together by polysaccharides which are produced in dictyosomes. 30. What are the most abundant components of a plant cell wall? How do the components interact, and how are they arranged with respect to each other? What are the symplast and the apoplast in plants? 31. What is the type of cell wall present in all plant cells? What is the type present in certain cells that must be unusually strong? 32. What are the extremely fine holes that interconnect plant cells? What is the name of the especially thin areas of walls where these holes are particularly common? 33. What is an intercellular space? Diffusion through a gas-filled space is approximately times faster than through a liquid-filled space. Living cells deep inside a bulky object such as an apple need oxygen. Do you think the oxygen could diffuse to those cells fast enough if there were no intercellular spaces? Would you guess that all or most of the intercellular spaces are interconnected such that oxygen could diffuse deep into an apple without ever having to dissolve into protoplasm? Design credits: Hummingbird, Tongo 58 Moment Getty, Green Plant Cells, Shutterstock Incorporated Natalia Ora, Purple Tulip, Shutterstock Incorporated Marie C. Fields, Dandelion, Shutterstock Incorporated Daniel Kreisel, Poppy, Shutterstock Incorporated Saryuri, Plant Icon, Shutterstock Incorporated Vector, Digging Man Icon, Shutterstock Incorporated Z-Art. Chapter 4. Growth and Division of the Cell. Chapter Opener Image. The bodies of both plants and animals grow as their cells and nuclei divide and multiply. Almost all cells multiply by mitosis, such that every cell in the body has the same sets of genes as every other cell. This is true of most parts of flowers as well, but some of the cells of the stamens and the carpels multiply by meiosis, such that the reproductive cells each have only one set of genes. Prickly poppy, Argemone. Outline. Box 4 to 2 Alternatives, Rates of Growth. Box 4 to 3 Botany and Beyond, Chloroplast Division during Leaf Growth. Learning Objectives. After reading this chapter, students will be able to state the 2 phases of the cell cycle. Order and explain the 3 phases within interphase. List the 4 phases of chromatid separation during mitosis. Explain mitosis and meiosis. Order the 5 stages of prophase I of meiosis. Summarize the cell division process in algae. List one difference in the cell division process between prokaryotes and eukaryotes. Did you know? The processes of nuclear division are almost identical in plants and animals. The processes by which plant cells divide differ greatly from the processes used by animals. Cells in mature plant organs such as fully expanded leaves or open flowers typically do not divide, only cells in very immature organs divide. Mitochondria and plastids replicate their DNA and divide using processes similar to those of bacteria. Concepts. The life cycle of individual organisms includes stages of initiation, growth, and death. Individual cells also have a life cycle, the cell cycle. Cells are initiated by division of a mother cell, grow for a period, and usually cease to exist by dividing and producing two daughter cells. This is not a real death because the substance of the mother cell continues to exist in the daughter cells. As a multicellular organism grows, this type of cell cycle is common, but as parts of a plant reach their final form, most cells stop dividing and enter an extended period of growth, during which they differentiate and mature. For example, leaf cells stop dividing when the leaf is only a few millimeters long. They continue to grow as the leaf expands and then function for the rest of the leaf's life. Leaf cells cannot normally be stimulated to divide again. If the leaf is damaged, little regeneration is possible. In contrast, cortex cells in the stem or root also stop dividing before the organ is mature, but if damaged, the remaining cells resume division and remain active until the damaged portion has been protected with a layer of cork. Some cells live for many years, even hundreds of years, but others die shortly after they mature. Bark cells are more protective if dead. Many flower parts die only a few days after the flower first opens, and gland cells often die after a brief period of secretion. Some cells never stop dividing. Cells in the growing points at the tips of roots and shoots are constantly cycling, as are those that form wood and bark. When a cambial cell divides, one of the new daughters becomes a wood or bark cell, and the other remains part of the dividing layer. The wood or bark cell divides a few times and then differentiates, matures, and usually does not divide again. The daughter cell that stays in the dividing layer grows back to its original size and divides again. Figure 4:1 The cell cycle consists of division and growth phases. The cell has grown and is ready for division. First the nucleus divides, and then the cytoplasm is divided by the formation of a new wall. The new cells enter a new cell cycle by growing. Then one divides again, but the other may begin to differentiate. In this example, its wall thickens as it matures into a fiber. The upper cell has finished dividing, and both daughter cells may enter new cell cycles. The lower cell continues differentiation. In some fiber cells the nucleus may divide once or twice, but usually once fiber differentiation begins, the nucleus never divides again. A cross-section of a set of cells similar to those depicted in the red cells have stopped dividing and are differentiating into fiber cells. The blue cells with thin walls are still capable of growth and cell division. Figure 4:2 Palms are extreme examples of plants that have only a few dividing cells. In this entire palm shoot, cell division only occurs at the extreme apex, in developing leaves, and in developing flowers. Of all the millions of cells in the visible trunk here, none is dividing. However, most cells are still alive. Cells at the base of the trunk were produced when the plant was just a seedling and have lived for about a century. Figure 4:3 Cells in this geranium bark divide only a few times and then differentiate by placing antimicrobial compounds in their walls along with waterproofing compounds. The cells then die and degenerate to cell walls without protoplasts. Lack of a protoplast is selectively advantageous because if a fungus or insect does penetrate the wall, it encounters no nutritious protoplasm and starves. Figure 4:4 In contrast to palms, this conifer has many groups of dividing cells. Each bud has a group of dividing cells at its apex. The needle-shaped leaves about to emerge probably formed most of their cells while enclosed in the bud and will now just undergo cell enlargement and maturation. Inside the woody brown stem is a layer of dividing cells that will add more wood and bark cells, making this part of the stem wider but not longer. Figure 4:5 The growing shoot tip of a cactus. All of its cells are alive and undergo repeated cell division. One of the basic roles of shoot tips is to convert nutrients into new cells that can later differentiate into shoot tissues. At the base, cells are lightly stained because their large vacuoles do not absorb the stain. These cells cycle more slowly than the smaller, less vacuolate cells at the tip. The cell cycle can be divided into a growth phase and a division phase. Growth phase of the cell cycle. In the 1800s, when the cell cycle was first being studied intensively, researchers gave the greatest attention to the division activities because many events could be identified. They assumed that between divisions cells were resting, so the growth phase was called the resting phase, or interphase. We now know that the cell is active during interphase, and it is possible to detect three distinct phases within interphase: G, S, and G. G phase. In G, the first stage after division, the cell is recovering from division and conducting most of its normal metabolism. One important process is the synthesis of nucleotides used for the next round of DNA replication. The length of a cell cycle varies tremendously, depending on the type of cell, the type of plant, its health, its age, the temperature, and many other factors. In single-celled organisms such as some algae, the cell cycle may be as brief as several hours. Short cycle times also occur in rapidly growing embryos and roots. On the other hand, cell cycle times of 2 to 3 days or even months are not unusual in tissues or plants that grow slowly. During winter dormancy, the cell cycle may last from autumn until spring. When dormancy is broken and metabolism accelerates, the cell cycle may last only a few hours. The actual length of time that any cell spends in G is similarly variable. Despite exceptions, in general, G is the longest part of the cell cycle. After a cell undergoes cell cycle arrest, stops dividing, and begins to differentiate and mature, it may enter a state similar to G and remain in it for life. Figure 4:6 Comparison of cell cycles of different tissues or species. In a shoot tip, cell division may occur as soon as the cell grows to its proper size, and G is only a small part of the cell cycle. Below the shoot tip, cells still divide but spend more of their time growing and carrying out non-division metabolism. G is a greater proportion of the cell cycle. Both and give only the relative amount of time spent in each phase, not the absolute time in hours or days. The cell cycle in cells like is often much longer than in shoot tips. S phase. During S phase, the genes in the nucleus are replicated. A gene is a polymer of nucleotides, and each gene has a unique sequence of nucleotides. Although many genes may have similar sizes, different genes never have exactly the same sequence of nucleotides. Many higher plants and animals need about 30,000 types of genes to store the information required to make the proper enzymes, structural proteins, and hormones necessary for the organism's life. The entire complex of genes for an organism is its genome. Obviously, if each individual gene floated around the cell or nucleus as an independent piece of DNA, then during division, it would be extraordinarily difficult to find each of those 60,000 genes and make certain that each daughter cell received one of each. This is not a problem, however, because all genes are attached to other genes by short pieces of linking DNA. Thousands of genes are attached together in a linear sequence. The entire structure is a chromosome. It might seem reasonable for all genes to link together in a single chromosome, but such a long chromosome would probably become hopelessly tangled. Only a few plants have as few as 2 chromosomes for their genome. Most have between 5 and 30 chromosomes. Table 4: 1 cell cycle phase durations in root tip cells and cell cultures. Note that when carrot cells are grown in cell culture, the cell cycle becomes much longer, mostly because of G, which goes from being only 17% of the cell cycle in roots to 77% in cell culture. Figure 4:7 If each gene were a distinct piece of DNA, ensuring replication of each would be difficult, and ensuring that each daughter nucleus received one copy of each would be even more difficult. Grouping the genes into chromosomes makes them easier to manipulate. Here only 2 types of chromosomes are shown, long ones with genes 1 to 15 and short ones with genes 16 to 25. Figure 4:8 Each nucleus in the cells of Arabidopsis thaliana has 2 of each of these 5 types of chromosomes, one set inherited from the paternal and one from the maternal parent. The slender area in each represents the centromere. The lines labeled MT and CHL represent the DNA circles of mitochondria and plastids. the entire genome has been sequenced, we know which DNA nucleotide occurs at each site in each chromosome. Even with 20 different chromosomes, an organism with 30,000 genes would have an average of 1,500 genes on each chromosome, resulting in long pieces of DNA that might break if unprotected. In onion, the DNA in each nucleus is 10.5 meters long when all DNA molecules are measured. In lilies, it is 21.8 meters long. In eukaryotes, a special class of proteins called histones complexes with DNA and gives it both protection and structure. Chromosomes also have another structural feature, the centromere, which is usually located near the center of the chromosome. Each end is capped by a telomere. Table 4. Number of chromosomes in a haploid set. A haploid set is a single set of chromosomes, as carried by sperm cells and egg cells. A fertilized egg has 2 sets and is diploid. Figure 4. 9 histone proteins associate into short cylinders and then DNA winds around each cylinder. The DNA histone particles then associate into a compact arrangement. Figure 4. 10. This chromosome was taken from a cell during division. It had gone through S phase of the cell cycle, so it has 2 chromatids, not just one. The chromatin has coiled tightly and is visible as loops. Condensation has made the chromosome short and thick, and it can be moved around the cell easily. The pinched region in the center is the centromere. Figure 4. 11. Before S phase, each chromosome has 1 chromatid and 1 copy of each gene. After replication in S phase, each chromosome has 2 chromatids and 2 copies of each gene. The constriction represents the centromere. During S phase, linking pieces of DNA as well as genes are replicated, and new histone molecules complex with the new DNA. Thus, entire chromosomes, not just DNA, are replicated. After replication is finished, the duplicate DNA molecules remain attached at the centromere, connected by a protein called cohesin. Although it would be justifiable to call this a double chromosome or a pair of chromosomes, it is also called a chromosome. Now that we know how a chromosome changes during S phase, we call each half of the doubled chromosome a chromatid. After S phase, each chromosome has 2 identical chromatids. Before S phase, each chromosome has just 1 chromatid. A chromosome after S phase is twice as large as it was before S phase. Although many cells stop in G when they cease dividing and begin to mature, most plant cells enter S phase and replicate their DNA before they begin to differentiate. This may involve just a single cycle of DNA replication, resulting in a nucleus twice as large as would be expected, or it may continue for many rounds of DNA synthesis and the nucleus becomes gigantic. This process is endoreduplication, estimated to occur in 80% of all maturing plant cells. The resulting nucleus has many copies of each gene, as many as 8,192 copies in some cells of kidney beans. Endoreduplication occurs most often in hairs, glandular cells, and other cells that must have an extremely rapid, intense metabolism. The normal complement of 2 copies of each gene does not seem to make messenger RNA rapidly enough for such active cells. Such cells are also rich in ribosomes and produce large amounts of protein. Gene amplification is similar to endoreduplication, but only some genes are repeatedly replicated. The amplified genes are those needed for the specialized metabolism of the mature cell. For example, as a protein-rich seed develops, its cells need large quantities of the messenger RNA that codes for the storage protein, and those genes are amplified. However, it would be a waste of energy and resources to replicate all of the other genes as well. The most extreme case of gene amplification known is an era maculatum. Each nucleus has enough DNA for 24,576 copies of every gene. We do not know how many genes are being amplified and how many are present, as only 2 copies per nucleus, and thus, we cannot compute the actual number of copies of each amplified gene. However, it is certainly more than 24,576. Figure 4.12. In this root of Serpis, cells in the central column have enormous nuclei that have undergone endoreduplication. Each nucleus now has many copies of each gene, so they can produce messenger RNA very rapidly. Consequently, the cytoplasmic ribosomes produce protein quickly as well. Table 4, 3 examples of endoreduplication and gene amplification. C is a symbol that represents the amount of DNA present in a nucleus. A normal body cell has 2C before S phase and 4C after S phase. Values that are a power of 2 represent endoreduplication, those that are not represent gene amplification. Figure 4, 13 egg cells of pines and other conifers become enormous. Their nuclei are gigantic due to having large amounts of RNA. Like any egg cell nucleus, this must fuse with a sperm cell nucleus before it can divide. G phase. After S phase, the cell progresses into G phase, during which cells prepare for division. This phase lasts only about 3 to 5 hours. The alpha and beta tubulins necessary for spindle microtubules are synthesized, and the cell produces proteins necessary for processing chromosomes and breaking down the nuclear envelope. In cultured animal tissue, if a cell whose nucleus is dividing is forced to fuse with one in G, the second cell's nucleus also begins the first steps of nuclear division. This is evidence that during G the first cell produced factors necessary to start nuclear division and that these factors are located in the cytoplasm. G, S, and G constitute the interphase portion of the cell cycle. After G, division can occur. Division phase of the cell cycle. The actual division involves 2 processes: division of the nucleus, called karyokinesis, and division of the cytoplasm, called cytokinesis. There are 2 types of karyokinesis, mitosis and meiosis. Mitosis. Mitosis is duplication division. It is the more common type of karyokinesis, the method any multicellular organism uses as its body grows and the number of its cells increases. It is also used by eukaryotic unicellular organisms when they are not undergoing sexual reproduction. Mitosis is called duplication division because the nuclear genes are first copied, then one set of genes is separated from the other, and each is packed into its own nucleus. Each daughter nucleus is basically a duplicate of the original mother nucleus and a twin of the other. Mitosis produces nuclei that are more or less exact copies of the original nucleus, except for occasional errors. As mentioned, thousands of genes are linked together into just a few chromosomes, making separation of gene sets much easier. It is necessary to transport only one of each type of chromosome to each daughter nucleus. This is made even easier because the 2 new chromatids remain attached together by their shared centromere as the chromosome is replicated in S phase. It is necessary only to make certain that one half of the doubled, large chromosome goes to one end of the cell and the other half to the other end. If that happens with each chromosome, each end of the cell automatically receives one full set of genes. The mechanism that ensures this orderly separation of chromatids consists of the following 4 phases. Prophase. During interphase, the DNA of a chromosome exists as a long, extended double helix associated with histone protein. This open configuration allows enzyme complexes to find specific genes that must be read for the information they contain, but in this condition, a chromosome may be several centimeters long. It could be wrapped around the cell hundreds of times, making it impossible to pull one chromatid from the other. During prophase, however, the chromosomes condense, they undergo a change in the way the histones associate. Chromosomes begin to coil repeatedly, becoming shorter and thicker each time. In addition, a protein framework develops to which DNA binds. Chromosome condensation continues until chromosomes are only 2 to 5 micrometers long. In this form, they can be moved around the cell more easily. In this condensed form, we can actually see the chromosomes. The uncoiled molecules of interphase are too narrow to be resolved by light microscopy. As chromosomes condense and become visible during prophase, other events occur as well. The nucleolus becomes less distinct and usually completely disappears by the end of prophase. The nuclear envelope breaks into numerous vesicles, each attached to rough endoplasmic reticulum. In those organisms that have a set of centrioles, the daughter sets that were duplicated during the previous interphase now migrate to opposite poles of the cell. It had been thought that they simultaneously acted as microtubule organizing centers and produced a long set of microtubules, the spindle, between themselves, but even if centrioles are destroyed by laser microbeams in animals, the spindle still forms. It could be that the centrioles themselves are separated by spindle formation, and the association is a mechanism that ensures that each daughter cell receives a centriole. Figure 4.14 During mitotic nuclear division, one chromatid from each chromosome is pulled to one end of the cell, and the other chromatid of each chromosome goes to the other end. Details are given in the text. The spindle is composed of hundreds or thousands of microtubules. Some microtubules extend from one pole to the center of the cell where their ends overlap the ends of other microtubules that extend from the opposite pole. The two sets together, overlapping in the center, form a large framework. Other microtubules run from a pole to a centromere. The point of attachment is a kinetochore, a structure consisting of two layers of proteins, one layer bound tightly to centromere DNA and the other attached to spindle microtubules. Each centromere has two kinetochore faces, one attached by approximately 15 to 35 microtubules to one end of the spindle and the other face attached by a similar number to the other end of the spindle. Plants and people. Box 4:1 Controlled growth versus cancerous growth. The actual steps of karyokinesis and cytokinesis must be controlled if cells are to divide properly, but other aspects must be regulated as well. First, the rate and frequency of cell division are important in determining how rapidly or slowly one cell produces a mass of progeny cells. Second, orientation of both cell division and cell growth affects the shape of the growing mass of cells. If all cells divide with their new walls parallel to each other, the mass grows as a column, but if new cell walls occur in any plane, the mass grows in all three dimensions. The mass grows as a sheet if new cell walls can occur in two planes but not in the third. Finally, it is important to control which cells divide. If only some cells undergo cell division, they may produce a lump or outgrowth while the rest of the mass of cells remains unchanged. These factors are controlled accurately in plants. In a young embryonic plant, all cells divide, but later, cells at the tips of roots and shoots become centers of cell division and growth while the rest of the stem and root tissues mature and carry out their functions. By controlling rate, orientation, and location of cell division, plants produce cylindrical stems and roots, thin flat leaves and petals, and massive, three-dimensional fruits. Plants impose quiescence on certain cells and then reactivate them to growth and division later. Plants produce buds that are forced to remain quiescent for a long time, even years, but can then be stimulated by the plant to grow out as a branch or a flower. In young stems, epidermal cells grow rapidly enough to keep the stem covered, but then they mature and remain mitotically quiescent as they protect the plant. In many species, these can be reactivated years later, undergo cell division, and produce bark cells. Cell division and growth must be controlled in animals as well. During early stages of fetal development, all cells of human embryos divide and grow. Later, some cells divide more rapidly than others, but basically, most of our cells are mitotically active during much of our growth before birth. Then cells in certain tissues and organs undergo cell cycle arrest. They mature and never divide again, such as the cells of our eyes and our brains. Other cells never stop dividing and are active until we die, such as the layer of cells that produces our skin and hair and the bone marrow that generates most of our blood cells. Just as in plants, some of our cells enter a prolonged state of quiescence and later are activated to division. For example, surgical removal of part of the liver causes cells of the remaining portion to divide and restore the organ to an adequate size. Of course, this is not true of most of our organs. Some of our cells release themselves from cell cycle arrest and begin growing uncontrolled by the rest of the body. This is cancerous growth, and its severity depends on Which types of cells and organs are involved, how rapidly the cells divide, and whether the cells can migrate from their original site and invade surrounding tissues. It is well known that certain environmental factors act as carcinogens, agents that cause cancer by interfering with cell cycle arrest. Cigarette smoke is known to cause cancer of the lung and throat, and ultraviolet light triggers skin cancer. Whereas uncontrolled cancerous growth in humans may be fatal, it does not seem to be a problem in plants. Irregular lumps and growths, called galls, may occur, but these are often caused by insects or microbes, not by the plant's own cells undergoing a spontaneous, self-induced release from cell cycle arrest. It may be that plants do form cancerous growths, but that they are not a serious problem for several reasons. First, cells cannot migrate through a plant body the way that our cells migrate through our bodies. Consequently, any uncontrolled growth is localized, not invasive. Second, whereas we have many organs that are each critical to our life and which occur singly or in pairs, plants have many leaves, roots, and flowers, and no single one is indispensable. Damage to one part of the plant may have little effect on the rest of the plant. By lacking the highly differentiated, complex, and tightly integrated body of humans and many animals, plants are not so threatened by diseases involving control of nuclear and cellular division. Figure 4:15 Early in mitosis, spindle microtubules attach to chromosomes at the kinetochore of the centromere. Each chromatid is attached only to microtubules coming from one end of the cell. Other microtubules pass from pole to pole or overlap the ends of other microtubules at the center of the spindle. As the kinetochore-attached microtubules shorten, the two chromatids of each chromosome are pulled in opposite directions. The long microtubules that do not attach to chromatids act as a framework such that chromatids are pulled apart rather than having the spindle poles pulled into the center of the cell. Figure 4:16 Anaphase in hyacinth root tips. The gray material between the red chromosomes consists of masses of spindle microtubules. These roots grow quickly and cell cycle times are short, and thus, at any particular time, many cells are in mitosis rather than just in interphase. Metaphase. After spindle microtubules attach to centromeres, they push and pull on the chromosomes and gradually move them to the cell center. Their arrangement there is called a metaphase plate. Viewed from the side, the chromosomes appear to be aligned in the very center, but viewed from the end, they are seen to be distributed throughout the central plane of the cell. No distinct boundary occurs between prophase and metaphase. Chromosomes gradually become visible and gradually move to the metaphase plate. Fragments of nucleolus and nuclear envelope may persist well into metaphase. However, a distinct boundary is evident between metaphase and the following anaphase. At the end of metaphase, a protein-degrading enzyme called separase is activated, and it digests cohesin. The two chromatids of each chromosome are suddenly free of each other. In this step, the number of chromosomes is doubled, but the size of each chromosome is halved. Each chromosome is like it was in G, before S phase. Although the number of chromosomes in the cell is suddenly doubled, the doubling is transitory because the cell soon divides. Anaphase. Anaphase begins just after cohesin releases the centromeres, freeing the two kinetochore faces from each other. Spindle microtubules that run to the kinetochore shorten, depolymerizing at the end near the spindle pole, pulling each daughter chromosome away from its twin. Agents that inhibit microtubule depolymerization, such as deuterium oxide, also inhibit chromosome movement, whereas those that speed depolymerization, such as low levels of colchicine, speed movement. The energy necessary to move a chromosome from the metaphase plate to the end of the spindle is small, just 20 ATP molecules. Long chromosomes may tangle somewhat, but microtubules exert sufficient pull to untangle them and drag them to the ends of the spindle. Because the spindle is shaped like a football, as chromosomes on each side get closer to the end, they are pulled together into a compact space. Telophase. As chromosomes approach the ends of the spindle, fragments of nuclear envelope appear near them, connect with each other, and form complete nuclear envelopes at each end of the cell. The total surface area of the two new nuclei is larger than that of the envelope of the original mother nucleus. Extramembrane may be derived from endoplasmic reticulum. Chromosomes become less distinct because they start to uncoil. Gradually, new nucleoli appear as the ribosomal genes become active and produce ribosome subunits. The spindle depolymerizes completely and disappears. Most events in telophase are reversals of those in prophase. To summarize mitosis, after G is completed at the end of interphase, each chromosome has been replicated and consists of twin chromatids. Spindle microtubules from opposite poles attached to the centromeres then pull the twin chromatids of each chromosome away from each other. Two new nuclei form, each containing a full set of chromosomes and each chromosome having one chromatid. The new nuclei are identical to each other and to the original nucleus that began mitosis. The nuclei can then enter G of interphase. The steps of mitosis are part of a continuous process and intergrade with each other. Similarly, prophase and telophase intergrade with interphase. Cytokinesis. The division of the protoplast is much simpler than the division of the nucleus. Although it is necessary for each daughter cell to receive some of each type of organelle, random distribution of organelles in the mother cell usually ensures this. No matter how the cell divides, each half typically contains some mitochondria, some plastids, some ER, some vacuoles, and so on. It is not necessary for each daughter cell to get exactly half of each. A single mitochondrion can divide, or a fragment of ER can grow until the cell has an adequate amount. The same is not true for genes. If one daughter cell is missing a gene or chromosome, the other genes cannot regenerate the missing information. During prophase, a set of microtubules and actin filaments aggregates into a band running around the cell, just interior to the plasma membrane. This pre-prophase band identifies the plane of division, and it marks the region where the new cell wall will attach to the existing wall. If the pre-prophase band forms around the middle of a long cell, the cell will divide into two equal-sized cells, but if it forms at one end, then division will produce a large and a small cell. The pre-prophase band is transitory, and its microtubules quickly disassemble and are recycled into the mitotic spindle. If nuclear division occurs without cell division, a pre-prophase band is not formed. Cytokinesis in plants involves formation of a phragmoplast, a set of short microtubules aligned parallel to the spindle microtubules. Actin filaments are also present. The phragmoplast forms in the cell center where the metaphase plate had been. Phragmoplast microtubules trap dictyosome vesicles that then fuse into a large, flat, plate-like vesicle in which two new primary walls and a middle lamella begin to form. The phragmoplast grows outward toward the walls of the original cell. It may take half a day to reach the walls of large cells. Phragmoplast microtubules do not actually migrate toward the walls. Instead, new microtubules polymerize near the outer edge of the phragmoplast, whereas those on the inner side depolymerize. New dictyosome vesicles are trapped and added to the edge of the large vesicle. It too grows outward, following the phragmoplast. Similarly, the new walls extend outward along their edges. The phragmoplast, vesicle, and walls are called the cell plate. This process continues until the large vesicle meets the mother cell's plasma membrane, the two fuse, and the vesicle membrane becomes a part of the plasma membranes of the two daughter cells. Simultaneously, the new walls meet and fuse with the wall of the mother cell, completing the division of the mother cell into two daughter cells. Figure 4:17 Late telophase in onion. The row of small vesicles is the beginning of a cell plate. At the edges are short segments of microtubules, the phragmoplast. Cell division has been interrupted by the herbicide DCPA. Alternatives. Box 4-2 Rates of growth. An organism, or a part of an organism, can produce more cells in a variety of ways. The two ways that are most important for you to understand are called arithmetic and geometric increase. In arithmetic increase, only one cell is allowed to divide. Of the two resulting progeny cells, one continues to divide, but the other undergoes cell cycle arrest and begins to develop, differentiate, and mature. After each round of cell division, only a single cell remains capable of division, and one new body cell exists. For example, starting with a single cell, after round 1 of cell division, there is one dividing cell and one body cell. After round 2, there are 2 body cells, and after round 3, there are 3, and so on. To obtain a plant body containing 1 million cells, the plant's single dividing cell would undergo 1 million rounds of nuclear and cellular division. If each round requires 1 day, arithmetic increase would require 1 million days, or 2,739.7 years. As you can imagine, arithmetic increase is too slow to ever produce an entire plant or animal. However, it is capable of producing the small number of cells present in very small parts of plants. For example, the hairs on many leaves and stems consist of just a single row of cells produced by the division of the basal cell, the cell at the bottom of the hair next to the other epidermal cells. The hair may contain 5 to 10 cells, and thus, all of its cells could be produced in just 5 to 10 days by divisions of the basal cell. This would be quick compared with the rest of leaf development, which might take weeks or months. An alternative pattern of growth by cell division, geometric increase, results if all cells of the organism or tissue are active mitotically. Again, starting with a single cell, after round 1 of cell division, there are 2 cells capable of cell division.